Okay. Greetings and welcome to one and all. This is Pritika. I will be taking you through the second day of Golden Jubilee lecture series of Shastri Indo-Canadian Institute and jointly hosted by Dr. MJR Education and Research Institute. A special welcome to all our speakers of today's session who has accepted our request and has come here to share their knowledge with us. And there is a small announcement to the participants. Participants, please post your questions either in the Zoom chat box or YouTube link chat box. The questions can be with the speaker's name, so we could post it. I could read it out with your name and the speaker's name at the end of the final talk. So, as a continuation of yesterday's session, we have Dr. Dirk here with his first lecture for today on environmental analysis. I think we can move over to Dr. Dirk. It's stages for you.
So before that, can I just very quickly summarize what happened yesterday and then introduce?
Oh yeah, sure, ma'am, sure. Yeah, sure.
Just come in today.
Oh yeah, definitely.
Okay, so good morning to Dr. Dirk from all the way from Canada, and a good evening to all my participants and the speaker of the day, Dr. Archana. Nice to have you all back here. We started this 2-day session, lecture series on DNA barcoding and metabarcoding yesterday. We had 3 talks yesterday. I introduced the concept of DNA barcoding and how we plan to use it. Followed by that was Dr. Steinke, who gave a very elaborate talk, giving us details on barcoding and metabarcoding, comparing metabarcoding with metagenomics, telling us a lot of details, a little bit about the bioinformatics packages that are available. We can see that that's a science which is growing very rapidly, and we really look forward to your talk today. Followed by that, we had a talk by Dr. Sudha Rao, who gave us perspectives on metagenomics and some metabarcoding using bacterial species working on bacterial diversity. So with this, we are looking forward to the talks by Dr. Steinke and by Dr. Archana today. So just very quickly to introduce Dr. Dirk to those who may have missed his CV yesterday. Yeah, Dr. Dirk Steinke is an Associate Director Analytics in the Center for Biodiversity Genomics at the University of Guelph, Canada. He is an eminent researcher in the field of DNA barcoding and metabarcoding. He has published more than 80 research articles and has currently 3 research projects going, and he is a reviewer to very respectable journals in this field. And apart from his research career, he participates in academic roles, scientific blogs, and is a member of many research communities. Over to Dr. Dirk Steinke.
Thank you very much. And yeah, good evening to all of you. Your day is almost done. I have to start mine. So we're doing a little bit of time travel and going into the past. Yeah, this morning I'm talking about environmental DNA analysis. I'm trying something new today, just in terms of how this is going to be looking. Hopefully you can see that now with me somewhere in the corner. So when I was thinking about this talk, which is sort of an extension of what I was talking about yesterday, and you will notice that sometime I'm going to be going back to what I was saying about metabarcoding for good reasons. So I was really trying to think of something that is a mixture of what you use technology-wise, because this is what it's mainly about when you talk about eDNA these days, but also a bit about the special feature of something that's called environmental DNA and what kind of advantages that has, but also what kind of pitfalls you can fall into when you work with this. So in the next little while, I'm trying to introduce to you what the definition for environmental DNA is, how we are actually extracting it and getting it, and then later on, what type of lab work is possible with it, and then later on also a discussion on, yeah, where can you use it, where should you be very careful in assessing your results, because you will very quickly see that we're talking about something rather sensitive, and probably sometimes you would probably call it ephemeral, because it's a very hard to grasp phenomenon. So let's start with the definition. I'd like to get those out of the way very quickly. So environmental DNA is essentially genetic material that you directly obtain from the environment. And I show you the environments we're talking about in a little while, and then without any signs of biological source material. So it's free-floating DNA as much as other sources of DNA, but it's not attached to an organism. So it's pretty much the opposite in terms of DNA of what you usually obtain, a sample or a community sample, bulk sample, which was a lot what I talked about yesterday. So let's look at it a little more closer. So here you see on one hand, so on the left-hand side, you see some potential organisms that shed their DNA throughout their lifetime, which is a very normal process. Humans do that all the time. Actually, we shed our skin or the upper skin cell layers pretty much weekly, and leave skin cells behind everywhere we go. And this is essentially environmental DNA. So you can, with the right methods and enough carefulness, extract genomic information from such cells. So you pretty much can do a sweep and then in a room and try to obtain this genetic information of the people that have been in there. So DNA, environmental DNA, can occur in several stages. It can be still within a cell, so the skin cells are just referred to as one example, but those could be broken up, so there's already some decay happening, so cell DNA is freely available. This is more a process when you're looking at within water, where you can have all forms of environmental DNA, from bound to cells to other matrices, all the way down to already several levels of decay, so smaller fragments, larger fragments of environmental DNA. There's everything in there. And then you can imagine, that's sort of indicated here on the right-hand side of the slide, that all these little particles that have some form of DNA bound to them or just floating around there are subjected to a lot of transport mechanisms. So within water, they just follow wherever the water moves. It's the same for environmental DNA that's somewhere in a sediment that's transported across the surface or anything else, or even through air, there's particles moving, and DNA is one of them. So there's a lot of different forms of environmental DNA, or let's say stages of decay, from a dead cell loosened from a body, all the way down to small snippets of it. So there is something that Barnes and Turner have referred to as an ecology of environmental DNA. So we're looking at different things here. So on one hand, we're trying to assess what the origin of is. So is it just by digestion, excretion, secretion, exfoliation, or reproduction? So there's a lot of ways this DNA could have occurred. One of the probably most likely one is that an organism dies and slowly starts to decay or is decomposed actively by other organisms, and then slowly the cellular matrix is dissolved and then DNA is floating around. But there's lots and lots of other activities. As I just said, organisms shed, lose parts of the environment, they're injured. Reproduction releases very often gametes freely in a water column or anywhere else. So this exfoliation is clearly, plants lose their leaves all the time. So there's lots of different sources of natural occurring loss of DNA from an organism. And then the environmental DNA is free. And then its fate is normally that it's slowly, mechanically, and enzymatically degraded. There's also, if it's free floating, chemical degradation. There's a lot of substances that shear DNA very quickly. Radiation plays a role, and we're talking about natural radiation here as well, especially UV light. And Then we also look at the environmental DNA from the perspective of its state. So is it intramembranous, extramembranous, is it particular, is it free? So particular can be bound to cell material still, but it also can be that DNA is bound to another substrate, like sediment particles or anything like that, which gives it in some instances, actually a longer lifetime than it would be if it's free floating. And then, as I said before, we look at it from the perspective of how it's transported within the environment. So is it settling somewhere within a sediment? Is it diffusion? So is it diffusing somewhere to other places? Advection, resuspension, so resuspending DNA out of the, for example, the sediment in a river, which is constantly moved around. There's still some biological transport going on, or anthropogenic, so human transport, so we inadvertently probably transport eDNA further than it would be in a natural way. So how do we make use of that? Because the idea is that all these free floating DNA contains valuable information. And since we are so good these days in obtaining sequences, DNA sequences that is, so we have been started thinking already for a couple of years how to make use of this environmental DNA. And I will talk about some of the potential uses later on in this talk, and then certainly also touch on it in my second talk this morning or your evening to show you what it's been used for. Generally, when you look at the sources of it, where we would go collecting and try to find our environmental DNA, you certainly look at 4 different general areas where you would look at. The first one is sediment or soil, where you can actually extract the DNA by probably washing it out. We'll talk about that in a minute, how these methods work, but we're essentially looking at people taking sediment samples within water bodies, soil samples everywhere in the environment. So this could be soil cores. You could use a dredge for doing that. There's lots of different methodologies out there in order to obtain a soil sample. And that's been done even without looking at environmental DNA in particular in the past. So either you sieve out your eukaryotic organisms, or, and that brings me to something that I should mention, there is a bit of within the definition of environmental DNA, there's a bit of an uncertainty when it refers to single cell organisms like prokaryotes, bacteria. They are part of the water column, or any kind of sample you're looking at here, or here in this case, the soil and the sediment. They fulfill the definition of environmental DNA. They're still bound to one cell, but also bound to a fully functional organism. So some people include them in the definitions. Many people say no, because the organism is still functioning and probably even actively moving around. So they say this is not really environmental DNA, but they are a reality when it comes to all your methods in the lab, because essentially when you're extracting environmental DNA and you're targeting, for example, cellular bound environmental DNA, then you certainly get all the bacterial DNA as well, and then probably consider it contamination, unless you are a microbiologist who's actually actively looking for that. And in particular for soil, there's a lot of work done in relation to soil microbiomes, in relation to agricultural research, where you're trying to obtain the bacterial communities. So as such, you could talk about that as a bulk sample of bacteria. So sediment, soil, water samples, I've touched on that also before in my talk about metabarcoding. So there's a lot of different ways you can obtain those. You can actively sample that with the pump systems you see here in this little collage. So there's certainly there's bigger and smaller ones. Some you can carry around as a backpack and have a little thing in your hand where you can obtain a sample. There's a filter pump built in. There's smaller versions now out there, which they call citizen science samplers. So they're rather cheap and can be handled by pretty much everyone, sort of involving the general public in your research. And you can also go and very simply take a bucket of water or another container and bring the water sample with you, and it contains a lot of eDNA. Other things we're looking at is biofilm. So you really scratch certain surfaces where organisms have started colonizing, and then there is a lot of decay products and environmental DNA within them. So you could think of any kind of drainage pipes where you scratch in the inside and trying to figure out what's going on there. Biofilms have been initially with metagenomic methods been looked at when they came out of caves where people scratched on rocks and everything. So you can think about the fouling on ships. We scratch up ship surfaces that have been long in the water and things like that. And another, probably some people not all of a sudden think about that, but air is also a good source for environmental DNA. The only issue there, talking about issue, is really just having the right sampling method to obtain enough material, because you could think that in air there's a very low concentration of them. But there's proper samplers. So we're talking about, for example, pollen that flies around and other, again, DNA molecules, although within air they're probably not as stable as they were in probably a solution with water, like in the water column or in a sediment. So those are the general sources, and then you can go very specifically what we're looking at, what kind of type of organisms you can find in all of them. So if you look at water, that it can all the way go from pretty much all eukaryotes and prokaryotes that live there. So we're talking plankton, metazoans, protists, plants, fungi, fish, amphibians, reptiles, and a few other things you probably not initially would think about. One of them I'm going to touch onto pretty much at the end of my next talk, just to show you how much you can push the limit of environmental DNA in there. And there's lots and lots of different things, but you can see there's a variety of organisms distributed in all these different environments. And people have started to try out pretty much everything these days. So from the early days of somebody having an idea that environmental DNA might be a good source, we're now moved ahead into the territory where research mainly focuses on two aspects. Number one is the more technical and the ecology of eDNA. So how does it behave in this particular environment? And with that connected is, of course, the idea that we get a better understanding of what the signal we are obtaining through our methods actually really means and where its boundaries are. And then the second one is still this little try-out phase where people try to find out how far can you push the limit of this? Where's the detection limit? Where can you find meaningful eDNA information in whatever sample you could possibly imagine? So we're doing both at the same time, but I think from a scientific and practical standpoint, it's done the right way. So you're really trying to find out how good is your method? How good is it in quantification? How sensitive is it? And also we're trying to see how you can proper validate these kind of procedures, because it's starting to be used very, very often in regulatory contexts. And there you need a proper validation. You need proof of concepts there in order to be able to really use them in regulatory contexts. So, wow, let's go to that one point where we're looking at how to get the DNA out of our sample. Because I've talked about in the metabarcoding part about we take bulk samples in various different ways. And to some extent, this is similar, but we have to have methods that actually extract the DNA first out of its medium, out of its environment, probably the bound particles it comes with and all that. And that's slightly different from dealing with a bulk sample. As you remember, a bulk sample is either homogenized as such when you do metabarcoding, or when you want to keep the organisms, and you go directly into lysis, or you take the preservation and try to extract the DNA from it. And that is the point what I'm trying to make. When somebody takes a metabarcoding sample and says, I want to keep everything in there untouched, but I take the ethanol out of it and try to extract DNA from that. That is essentially a use of environmental DNA, because that's a DNA that's released from all the organisms in this sample. And it's already in ethanol, and then you go further with it. And one method of doing that, and one we've been doing in our lab, is actually filtering. So that would be referring to what you see on your screen with filtering in the laboratory. So where you have certain devices, either it's a really simple filter which is commonly used. Or if you have larger quantities, you very often have filters that are put on vacuum pumps, which suck the liquid out, and then you have the filter really purely there. The other way is, of course, filtering out in the field. That's what I was referring to before with the people with these backpacks on their thing, or this citizen science samplers. These are essentially pumps that come with a filtering system. And there's a lot of development currently going on in terms of how to optimize these pumping mechanisms so that you don't have to carry around big batteries or fuel or whatever to maintain your pumps. You actually sometimes don't need a pump at all. So you could do that with a larger syringe and a filter attached to it. And then you just push water through repeatedly, pretty much until you can't do it anymore because your filter at some point clogs with everything that's in the water and you just push through it. And if I talk about filtering, it's not only the water, it's also filtering we use when we do sediment cores, because the first thing we do is mix them with some liquid medium in order to filter them through. And even the aerial samplers use filters to some extent. So it is not such one method that's focused on this one environment, water. It's all the others too. In the field, you can have filters that are that need some sort of preservation chemistry, but there's newer filters out there, although they're not necessarily cheap, that are self-preserving. So essentially, you use them and then you pack them in a bag and you can carry them home, and they're fine. They keep the DNA intact, and also they're preserved for a while, so you don't have to worry about bringing any kind of extra preservatives with you in the field. I'm pretty sure you can't see me anymore because I think somebody is not muted. Is that correct? Can you see me, or can you see my presentation?
Yes, sir. We can see your presentation. Yeah.
So
okay, that's fine.
Yeah, it should be fine. Yeah.
Okay. Just let me know if there is a problem with that. So if you can still see the slides, that's fine. Otherwise, I'll have to switch. So self-preserving filters is the newest thing there, and you really just bring them home. And from these filters on, you have all the methods at hand in order to do further regular extraction protocols. So there's various protocols of them. Very many times people just take the filter, cut it in small pieces, they dry them or grind them up, or there's ways. There's particular filters, if you use them, you can actually dissolve them in acetone and things like that. So you're starting to use standard procedures once you have your filtrate. The other method is when you take a pure water samples, and very often that's the only way you can do things. You can't bring the big pump or whatever, or you have smaller quantities of liquid that you can bring or sample, because a big filter pump easily goes to tens of liters in order to obtain enough for the filter. But if you don't have that, then you will have to go and use the water sample as such. And then one method to do that, the filtering is probably not sufficient, because there's a certain amount that will just go through the filter pores and will not be helped by the filter itself. So it's better to just make, and if your DNA concentration is low, that means you would lose even more. So the alternative method is really just going through a precipitation thing. There's very easy cold ethanol precipitation methods out there and a few modifications thereof. They work quite fine, as I said, especially when you have low concentration environments. This is the method to go to. And also when you have only small quantities of sample, you can actually use, and where filtering wouldn't get you anywhere. And of course, you can go in a way through the homogenization procedure. This is very often done, or a method pretty much copied from the bacterial research when they work with soil and everything else. So you suspend them, then you use beads or anything to shake it up. So it's very similar to a bulk sample. You want to homogenize, and then the downstream procedures are very similar. So now you have obtained your DNA. You have it extracted with one of the standard methods. And yes, once you have your filtrate ready, you can use any kind of kit that is out there or homemade methods. It should pretty much give you the same information. So now we're coming to the point where work can deviate or where things can split up depending on what you want to do. There's two approaches to use of the eDNA. One is you know what you're looking for, what means you have a targeted assay in mind where you say, I would like to use eDNA to find an organism of which I don't know whether it's in that particular environment or not. That's the biggest question you're asking there. For that, you don't need to go down the sequencing path. You don't need to do metabarcoding because you have one single organism or probably a few. You can multiplex these things to some extent. So you have a target organism in mind. This is very often used these days either in conservation, where people are interested in whether certain endangered organisms that are hard to be observed, where you don't know whether they live in a certain place or not, where you go out there and take samples, environmental samples, and try to detect if they're still occurring there or not. So they're extending our repertoire, which so far was observation, by using environmental DNA to do that. Very helpful in conservation issues, as I said, but also in another area, and I know here in North America, very often the eDNA use is actually for detecting invasive species. So we have problems with certain species all over the place that are inadvertently were imported, or actually sometimes not only inadvertently, people have deliberately introduced them without thinking what can happen down the road. And now we're trying to determine how far they already moved within the country. For us here, a very famous example is that in the US, people had been introducing a number of years ago 2 Asian carp species. They were for game fishing and other purposes, and they released them in some smaller lakes, but unfortunately these were connected to larger water bodies. And these species are very good in adapting to Canadian or North American environments. So they moved out there, they reproduced happily, and started to push out the local species. And they move in further into our Great Lakes. And a number of years ago, when eDNA methods with these target assays were brand new, researchers in the US went to Lake Michigan, took water samples, and determined that the species had already moved in there. Initially, people thought they couldn't be that far, but they were. Subsequently, all the way till today, we've seen that they moved into our Lake Ontario system, Lake Erie. So we know they're in the Great Lakes, and we're using these kinds of methods to determine how far they got. Because these lakes I'm talking about are rather big, so you're not going to do by chance go out there and try to fish the traditional ways and even electrofishing or so. You probably wouldn't obtain them. You only see them even jumping out of the water when the population is very, very dense. So we have a system that can detect both species of Asian carps, and what it uses is a target-specific assay, as you can see it in the slide. And the idea is that you actually design a little probe, which is a sequence that perfectly matches your target. So the DNA of one of the Asian carps needs to be 100% matched to this probe part. So this is the small sequence. At either end of the sequence are 2 different things attached to it. One is the reporter, which is a bioluminescent probe, although when it's bound to that sequence, to your probe, and at the other end, there's another thing called the quencher, which essentially does what the name says. It quenches the bioluminescence. So together, the 3 parts, as long as they hold together and both of the ends are very close together, so the probe should be short, nothing happens. No fluorescence appears, no light is excited. So when this probe binds during PCR, and we're using something that's called quantitative PCR in this case, or real-time PCR. So when you run a regular PCR, the probe binds to your target DNA. At the same time, the polymerase started its normal PCR job, trying to elongate the fragment and try to do polymerization there. By the time it reaches the fragment, that's something that you see in the second of these little cartoons on the slide. It starts to digest or to cleave all these little parts of the probe itself. So the polymerase moves over there, and all of a sudden this little probe starts to fall into pieces, which means it releases the reporter. As soon as that happens, the reporter starts giving a signal out there. So a fluorescence that can be detected through the qPCR machine. And that's how you do this, because as soon as you get one of those signals, you know your probe bound to the right organism. It's also called real-time PCR because it happens really fast and in real time, and you can see, and what you obtain at the end are so-called curves that show you exactly a slow start and then how these fluorescences occur across the entire group of molecules in a higher level, and then it plateaus at some point when all the probes are used. And these curves are very characteristic for a particular organisms and reactions, so you can really determine what happens there. It also allows you, if you do proper calibrations initially, to determine the abundance indirectly through that. So here's a method that is more advanced than metabarcoding is at this point. You have a measure of abundance, but again, we're talking about a single species, not about an entire community. So this is how these little probes work. There's several of them. Probably most famous commercially sold one is Tacman. And this is what was used initially with the very first eDNA study that was published. And you're looking at this paper here in Biology Letters from 2008. So about 13 years ago, they came out with that one. Here, what they tried to do is actually use water samples from lakes, ponds in France, in Europe, to get an idea whether this wonderful frog you see here in the corner, probably a little hidden by my own image, but this little frog there, it's called a bullfrog. I hope you probably heard about these little guys, or not little, they're really good. They could be football sized, roughly. And these frogs do not belong to Europe. That's why they were interested in that. So that's a species that occurs here where I live naturally, but not in France. So this was the first study really doing a targeted approach, looking for an invasive species, because yes, you can hear these guys, but sometimes it's hard to find them, especially when they're not in their active period, and then you really have to go fishing. And the best idea is using water samples and use eDNA. So this was the first study done there. So that's the first way of using eDNA, targeted. That requires that you know what species you're after, and usually allows you only to do a few species at a time at most. And probably the most important part, we'll talk about it later, is study design there, where you spend most of it. But it's certainly, when you do it on a regular basis, much cheaper than the other method I'm just going to talk about, because you skip the entire sequencing, which could be a very costly part. What costs here is the development of the probe. But as soon as that's out there and you have one, and a functional one that works very specifically, and you've been looking at everything, so what species I'm looking at, is there population differences that might be throwing my probe off in some cases? You have to be very careful in designing that, but once this is settled, you have a system that goes forward for a long while and for relatively low cost. The semi-targeted approach is one we've already talked about yesterday. That's the next one, but I just skipped one slide, which I shouldn't have, because I looked a little closer at how you design these probes, because that's a bit of it's a longer work. So you really have to take into account the phylogeny of the study system you're looking at, because probes, because they're so specific, can sometimes, I mentioned that before, bind to certain representatives of a species, but not to others. So wherever you look, you have to look at the population differences of your particular target gene, so that your probe can bind to them all if you're interested in that. If you're interested just in a particular population, same story. You have to find a probe That fits perfectly. And of course, it needs to be within a target region. And very often in barcoding, you try to use CO1 for animals, for example. So you limit yourself by saying, okay, I want to use CO1 as my target region. That's where the PCR will go after. Now I have to find a little region within my CO1 gene of this particular species that 100% bind doesn't vary too much, so that my probe has a chance to really tightly bind to that. So you use that, so you do sequence collection, you look at alignments to doing that, you do primer and probe design, you figure out if that works, if the primers function properly for this particular species, if your probe binds properly, if it doesn't bind anything else that's not your species, that's the other thing you really have to look, so you don't want to have too many false positives in your mix. And you try to test the primers, you validate them a couple of times. You can do that even by using synthetic DNA. So actually DNA that you, for that purpose, have been built. That helps you quantifying and give you an idea of the validation curves. And then you try it with your field samples. It's an elaborate process, but you do it only once for a particular assay. So, and now I come into my semi-quantitative approach, semi-targeted approach, which is metabarcoding. It's essentially what we talked about yesterday all the time. You use your environmental sample. Your source is not a pile of organisms and not a homogenized powder of insects obtained from a Malay trap. You take your environmental sample, you filter, and subject it to pretty much the same procedures as we do with a bulk sample. The critical part here is just have your DNA extraction method obtain enough DNA to go forward with all the other procedures we're talking about. And there's a lot of potential applications, or not only potential, but really used applications to eDNA metabarcoding. So we've seen people using them in ancient ecosystems, so where they go digging, archaeological digs. We had one case here in Canada where we actually went out, or some archaeologists went out and dug out old settlements. And they were not that old, so we're not talking ancient old, a couple of 100 years, early human settlements here in North America. They looked at their kitchen garbage, they dug that out, and small fish bones they found there. So they could, as far as identifying this is a fish bone, but they had no way of identifying what species. So we essentially took that bone, grinded it up, looked at the leftover DNA of this decaying, I think it's 800 years old, and found out what kind of fish species people were having for dinner 800 years ago. So you could look at, in this case, plant pollinator interactions. I'll talk about that in my other talk. Dietary analysis, same story. I have a few examples for that. You can detect invasive species as well, especially if you're trying to do a survey of what's really not belonging here. Or in one case, we, for example, do that sort of the other way around. Through our Malay trap programs, we have a fairly good understanding, especially for certain areas in Canada, what kind of terrestrial arthropods occur there. And that includes the invasive species we know about. But we also did a test at some of our harbor sites here in Canada, one in Halifax, so the Atlantic, and try to see at the point where many things arrive, where ship traffic is and brings in things on the ships, within the crates, whatever, containers. So we put a trap there and did metabarcoding and always comparing it to the reference library of what we know occurs in Canada. And sort of any time when a signal pops up that we don't know of, the next step would have been then take that signal and compare it to what's occurring in Europe. And if we find a match there, but not with us, we would contact our European colleagues that are experts for the particular groups and say, okay, is this an organism that could be of concern for this country? So does it survive our climate conditions? So is there a likelihood that it actually can establish itself here? Because normally when you have these kinds of organisms, only 1 in 1,000 probably has the capacity to stay there. But that one organism could be pretty bad, as we all know about invasive alien species and the damage they do and everything else. So this was a way of using also an eDNA sample then later on to see if we have new arrivals in a particular area. And of course, we have responses to pollution, can be measured directly. All of that can be measured also with traditional barcoding, in the sense, metabarcoding, in the sense that we use a bulk sample. But eDNA gives us probably the same idea. And of course, air quality monitoring, where environmental DNA is probably the only way to go forward. And here in this air quality monitoring, we're looking at biological air quality monitoring and not so much the chemistry and everything else that comes with it. So, some more thoughts on primers. Yesterday, I had quite a couple of them. And when it comes to metabarcoding of environmental DNA, there's some extra thoughts. Number one, yes, there's quite a few out there, and probably even more than we had for regular metabarcoding. But one important thing here is to consider. Because of the amount of degradation environmental DNA is already going through, you treat that sample of yours with an extra eye on primer development, because your DNA is largely already broken into smaller pieces. So it is probably of little use to go in there with the same primers you would be using for a metabarcoding of a fresh bulk sample. You're more looking at it from the perspective of what kind of fragments could I likely obtain there. So primers that are often used in eDNA studies look at shorter fragments. So, for example, if I'm using a primer such in these lists there, that is regularly for my next generation sequencing, probably just 400 to 500 base pairs long, the fragment that it retrieves is 400 to 500 base pairs long. Then if I'm doing environmental DNA, I would look at the top of my list where all of a sudden the fragment I'm obtaining is 150 base pairs only. That comes with a difficulty. The shorter your region is, the more you probably have unwanted amplifications or problems with matching that sequence to your reference library. So again, you have to be very careful in choosing the right fragment and testing again if it covers everything you want it to cover. So if you want a universal primer for a certain particular sample and you're trying to obtain as much information from it, you have to design one that is short and unambiguously matches that particular region. So you very often find eDNA primers ranging from 150 to 300 base pairs length. And that has to do with the fact that there is a decay process going on and you try to optimize the amount of things you can obtain with it. It is possible, many studies have shown it, but there's certainly an extra source of potential error in there. So I talked a little bit about abundance and some of the advantages of eDNA studies that they probably have over other traditional, they're not traditional, over metabarcoding anything, so we're still new there. So if we use qPCR quantification, and this is what this table should clearly get you as a primary take-home message, is the pro is you can easily obtain this, and the other one is you can get abundance to some extent. We don't know it for sure for all of them, but a couple of studies showed clearly there is a potential to get full, very good abundance measures. So they've done that for fish, where they had defined environments where they knew the abundance of the particular species, and they really looked at those assays. Of course, the approach to abundance that we know from metabarcoding, that you look at the proportions of reads assigned to a taxon in a sample, is a very bad one because of all the different biases that are involved. There's PCR bias, there's certainly the different orders of magnitude of biomass, so the introduction of how much DNA is in there. So it's not a very good measure. There's other people that said, you just look at the frequency of successful PCR amplification, of repeated PCR amplification. So you do it over and over again, and then you start quantifying how often was the particular organism detected in my different PCR approaches. The disadvantage of that is it costs a lot of money because you have to do a lot of PCRs. And it's probably worthwhile when you really have an important project in order to obtain that. And for some other reason, qPCR so doesn't work, but it's really something where it's not very economically feasible. It's a nice idea though, it works, but yeah. So the other thing eDNA samples allow you to go back in time. So I told you the example of the fish we found in the kitchen garbage of people that lived 800 years ago. Researchers have pushed it further back in the past, and in metagenomics even further. So you probably heard of some studies where they went to archaeological sites, Stone Age, I'd say, where researchers obtained sediment and debris from those areas. And this was a group in Leipzig that specializes on ancient DNA. So they were the ones that were working on the mammoth genome, on the woolly rhino, on the Neanderthal. And they also went in there and took environmental samples and reconstructed mitochondrial genomes of people from those debris. So this is how far you can probably go already. It's a very sensitive process. You have to be extremely cautious, especially when you work with human samples, because you can always, our own DNA on our fingers, if you don't work in a clean environment, our own DNA is going to ruin the entire analysis, because there's so much more active DNA on your hand than you would ever obtain in a huge sample from this region. So you have to be extremely cautious, have to be highly clean, which, by the way, is one thing that is important for most eDNA studies, because the concentration of DNA is so low, because fragments have been already fragmented more than you would like them to. You have to be way cleaner than you would probably do with all the other analysis. Contamination is a huge issue because it requires only a few molecules of intact working DNA in that sample to overshadow any signal you were looking after. This is particularly important if you work with human samples, but it's also important with everything else. Plus, all the enzymes we bring with us that are on our fingers can destroy a little DNA there even further. So you can track samples through age, which is this slide sort of symbolizing. So when you go from the left, from very old samples indicated by that mammoth there, you go to the more recent on the right. You can use eDNA in sediment cores to really do a little travel into the past and see what organisms lived there a couple of hundred or maybe 1,000 years ago. But the further down you go that route, the more fragmented DNA is, the more you have to be lucky that it's bound to some sort of a substrate which keeps it intact, so that the chemical conditions are good enough that the DNA molecule doesn't dissolve into its individual nucleotide pieces. So, and very often people also, that's why we go for bones, where some of the bone marrow inside is dried and we can still obtain some leftover DNA. In older studies, people go for teeth, things like that. It is possible. However, nothing is perfect. So here's some of the enemies of your environmental DNA in the picture. So you're looking at light and the radiation that can degrade DNA. Temperature can have a huge destructive influence, especially warmer temperatures. Time is always the enemy in that case, the longer things go on. And there's studies that try to find out how long free-floating DNA can persist in a particular environment. There's also microbes and free-floating enzymes out there that destroy the DNA. So for some microbes, it might be a food source. So there's a lot of things that work against it, so which is also making the concentration of DNA that's useful for your study a low one. And here's a few examples on how that can actually play out and ruin your result. So sometimes we don't know the sources of a peak in environmental DNA. So the first picture on the upper left shows you that you cannot distinguish between reproduction and decomposition when you see an eDNA signal. So if you spike of eDNA all of a sudden, it could be because there's a spawning event, which might be something you want to track. But at the same time, it could also be that there's some sort of a mass death event. So it's decomposition of many individuals going on at once. You trace only the eDNA that's released out there, and you see a spike in its concentration. But the sources for that are different, and it's very different in these two cases. Reproduction means there's an active live community. Decomposition means not necessarily there's any live fish around. Could be a lot of dead fish. So if you are trying to trace if there's fishes of that particular species living in that environment, you have to be very careful with your interpretation, because it could be that it's just a lot of dead fish. And that is not necessarily what you were after. The same goes very often when you have different speeds of decay. A slow decay and a fast decay of DNA might give you the same result to some extent. Or when you look at the state. So depending on what kind of filter you're using, you could get different eDNAs. So depending on how it binds to it, or what it is bound to. So if it's bound to certain sediment types, particle sizes matters a lot. So you might end up getting eDNA, not a lot of eDNA, not because it's not there, simply because it's bound in a, let's say, in a sediment substrate, in particles, and happily, cozily sitting next to these particles that are not caught by the method of choice. So it's just washed out in your filtrate or something else. So you're simply not obtaining it. It's more a technical problem, but it can be that because DNA, I wouldn't say bind, but it's closely associated with other microparticles, and more probably, we haven't looked at that. It's worth probably a study. Microplastic might play a role as well. So you can imagine that microplastic structures that are now everywhere could help eDNA sort of survive a little longer, but also probably just screwing up your entire laboratory procedure. And of course, there's transport. And DNA can, again, be transported with sediments someplace else. So your signal might not be a signal of this particular location. So you obtain a eDNA from a sample at point X, but that does not mean that the organism, the source organism for the DNA, was actually at this point. It could be transported through sediment, transported through the water column. If you're in a river system that's highly dynamic, so there's a strong current in there, DNA might be from upstream somewhere, and it's your job probably to initially determine how far can DNA travel in that system and how is it influenced by the different morphologies of the river. So it's not a very easy thing to interpret. If you take a sample at some point, it doesn't necessarily mean your organism is right there. It can be somewhere else. And very often it is. So here you can see some examples. So on the left hand, you can actually see the distribution of eDNA in a particular river and from a particular species. So it's a very dynamic system. So if there's DNA out there, it's not equally distributed. It depends on so many factors. And you see some of these factors in a river now on the right-hand side in that figure that shows you how it is transported in the water column by current, where it could be suspended and diluted and again downstream transported. Some of it is retained in the sediment, and then again sediment can be resuspended, so meaning there's a bit of turbidity in your system, then all of a sudden the eDNA floats out and is transported away to a more quieter area of the river or the lake or whatever, and then settles again. So then you could take a sediment sample of an area, obtain the eDNA there. It's not necessarily from just falling down or an organism dying on that area. It could be coming from somewhere else, floating around through resuspension. So there's a lot of hypotheses and lots of studies that are trying to look at eDNA movement in this regard. What else could we do, and what do people think about? So this is already my last slide, and this is the more speculative one, and partially people have started looking at it. So what about eRNA? That's, of course, a discussion where you could have to think about first the fact that RNA in general is usually even more unstable than DNA when it's free. But again, same as DNA, it can sort of associate itself with other particles. But what would be the advantage of the RNA? So there's a bit of the discussion of going on what's better, DNA or RNA in general. And I've talked about and touched about it in my metabarcoding talk. That RNA is actually a good indicator of certain stages. It's probably just a reflection of an active community. If you're looking at what's going on, although here we're looking at free-floating molecules. The questions that are mainly out there, and they're not 100% answered, is the question is, are eRNA a better marker for monitoring biodiversity, simply from a technical standpoint? Because as the same in Metabarcoding, we can obtain the RNA signal for all the RNAs that are out there, which allows us to dig into several different reference libraries that are maybe out there. This is probably more so for certain groups of organisms. We certainly do better with organism detection when it comes for animals, for example. With our cytochrome oxidase 1 barcode library is probably the largest for an individual marker. But for other systems, lots of ribosomal DNA and so on has been sequenced, so we can dive into that. And then the question is, what we wanted to do is rather, can you do broad-scale metatranscriptomics? So can you really look at the active set of all the organisms that are in this community you're sampling? So not an individual organism, all of them. And use eDNA samples or eRNA. It's very difficult to distinguish all the time, but use RNA samples to do that. Problem is the same. You have lots of degradation going on. They're pretty much the same phenomenon. There is persistence out there. We know that RNA is out there. It's floating around. It can persist through time. Its protective mechanisms are very similar to what helps eDNA, but again, it's more volatile, I'd say. The potential advantage of that one is, imagine our metabarcoding approach. We're going after a single marker. And we're going it in an environment where decay is one of the ruling factors. So the reason why CO1 as a barcode works in this regard is because of the high copy number. It comes from an individual organism. It's a mitochondrial gene. We have tons and tons of them in our cells. The same goes, and probably to a larger extent, for ribosomal, for RNA, because there's lots of it. We produce a lot of it. We produce messenger RNA, the rRNA in all our translational, transcriptional processes. It's all involved. So you have a higher likelihood of obtaining some information, which is critical in an environmental context when things are free floating and the concentration is low to begin with. There is probably a higher turnover than with eDNA. Lots going on there. And there are there's people who discuss that there are some protection mechanisms against degradation that help RNA better than it would help DNA. Not clear about that, so not sure. But one of the main advantages is that there's a high production rate, and there's good methods to actually obtain the RNA from the sample. We are in the business of testing that out. There's another laboratory here at McGill University with Melania Costescu who work a lot on that. It's really a time where this is the new thing where people actually play around with and see if it really works. We're not at a point where we say we recommend using that for everything else. It's not the day where we say, hey, we can replace eDNA, which we will never do, because it has proven to be such a good way of working with all that. So with that, I'm done.
Thank you very much, sir, for the informative lecture you gave us. Without any delay, I will call Ms. Aradhana, Research Associate of Dr. MGR Educational Research Institute, to introduce our next speaker of the session. Aradhana, over to you.
Thank you. We'll have questions at the end.
Yeah, we'll be having questions at the end.
Thank you, Kritika ma'am. Now, I would first like to thank Dr. Drake for giving us some insights on how to perform and understand about environmental DNA analysis. Now, I would like to introduce our next speaker of the day, Dr. Archana Chauhan, Assistant Professor in Department of Zoology from Punjab University, Chandigarh. Her earlier research career started as a PhD graduate at Institute of Microbial Technology, Chandigarh. Later, she completed her postdoctoral studies, which mainly focused on environmental biotechnology. She has published nearly 30 research articles in reputed journals. Currently, she is mentoring 5 doctoral students and many postgraduate students. Right now, her research focuses on biodiversity and climate change in regards with metabarcoding studies. In addition to it, she has 2 ongoing projects which deals with aquatic biodiversity and antimicrobials from bee sting. The topic for her today's lecture is monitoring of aquatic biodiversity using environmental DNA metabarcoding. Over to you, Archana ma'am.
Thank you. Thank you, Aradhana, Dr. Aradhana, for a very humble introduction. So I think we are a little behind schedule. So without further delay, I'll just share my screen, my presentation with you, and I'll start with my presentation. Okay, I hope you can see my presentation.
Yes.
Okay, great. So before starting, so I must say that Professor Dirk has already given a very beautiful introduction of the environmental DNA technology. And it was an excellent overview wherein he has almost covered most of the aspects and most of the basic and the important aspects related to the eDNA technology and metabarcoding technologies. So that is, I think it has created a good platform for me to go forward. So my talk basically would focus on as already told, would focus on the eDNA barcoding and metabarcoding. I'll not talk about barcoding, eDNA metabarcoding approach in the aquatic system. So when I say aquatic system, so I would not be talking in general, rather I would be specifically taking a case study wherein we have tried to use the eDNA metabarcoding approach to study the fish diversity. Okay, so that is what I would be, and the sampling site I would be talking about in detail about the sampling sites in a few minutes. Okay, so before I go further, the key terminologies associated or that will be used during the presentation will be indigenous. And indigenous, everybody knows, is a general terminology or scientific terminology which is used to for any species which is present in a particular region naturally. And similarly, exotic means that is any species, here it is for the fishes, which is not native to the geographical region, but it has been introduced into that particular region. eDNA, I hope now everybody knows what is eDNA, and NGS and HTS, again, I know that everybody is familiar with. So this slide gives you a brief overview of just a brief overview of how the eDNA studies have emerged from 1985 to 2015. So because we can say that technically the first mention of the eDNA or the environmental DNA extraction was mentioned in 1987. So this slide is just giving an overview of the same. Okay. So, though most of the aspects, they were covered by Professor Dirk, but I would also want to focus, and he has also emphasized on this, that most of the DNA which is present in the environment, specifically if we talk about the aquatic ecosystem, then the life of that DNA, environmental DNA, is not long. So if we, as published by DG et al. In 2011, we can just say that in a nutshell, the environmental DNA in an aquatic system on an average can have a life of 2 to 3 weeks. However, there is a standard deviation, or there is deviation in this depending upon the species or the organism we are interested in. So coming to the main presentation, so we know that fish diversity in India, or fish is a staple food in India, and therefore fish diversity in India is represented by 120 commercially important fish species, and out of which the 60 have been found to be very widely distributed in most of the water bodies. And in addition to this, there are 300 exotic species which includes 291 ornamental species, 31 aquaculture species, and 3 larvicidal fishes, which has also been introduced in India from the different areas. So the question is why they have been introduced. There are several reasons why the exotic fishes have been introduced into the Indian ecosystem, is aquatic ecosystem. The reason is very general: to increase the fish production in different water bodies, to broaden the species diversity in the local aquaculture. Some of the fishes have been introduced as sporting fishes, whereas some other fishes, they have been introduced for aquarium keeping. And while others, they have been introduced to control certain larvae, such as the mosquito larva or the other unwanted organisms. So, however, this was the aim for introducing the exotic fish species into the Indian ecosystem, but the problem is that it has been internationally recognized, and we have also seen in the Indian environment also that most of the introduction of the exotic species, fish species, has caused a lot of devastation of the native fish biodiversity, specifically in the freshwater ecosystems. The reason for this is because most of the exotic species which were introduced in the in the ecosystem or freshwater ecosystem, they had dominating characters. They were very aggressive. So, and they competed for food as well as space very naturally with that of the indigenous species. So this actually led to the alteration or extinction of the gene pool of the species stock. And not only by competition or dominance of the exotic species, but also by crossbreeding or hybridization. So the end result was that everything, because of this introduction of the exotic species, there was decrease in the number of the native fish species, or in some cases, there were even removal of the fish species from the natural habitat, the local fish species. So therefore, the exotic fishes are a global concern. And there have been a number of examples wherein the effect of introduction of the exotic species has been studied worldwide. For example, the effect of introduction of exotic fish species in Lake Banyuls, it was studied, and it was found that in 1910 there were 5 native species, but introduction of the 12 exotic species, as a result of that, 2 of the native species were completely lost, which was stickleback and tench, and there was decline in the number of the other 3 native species. So presently, this lake is now dominated by only the exotic species. Similarly, Lake Victoria, which is one of the largest tropical lakes, there were almost 300 endemic species contributing to 99% of the fish diversity of the lake. But then another exotic species, that is the Nile perch, was introduced. And again, the result is that most of the endemic species, they became extinct, and now they represent only 1% of the total lake's fish diversity. Similarly, blue tilapia. So it was introduced in the Lake Parker in Florida, 1968. And at that time, again, after 4 years in 1972, it was found that the indigenous fish, that is the gizzard shad, it reduced to 5% of the total catch, whereas blue tilapia, it was in greater abundance, contributing 68% of the total catch. So similarly, not only here, but in India also, this table, which is there on my slide, it shows that a number of exotic species were introduced in the Indian ecosystem also from the different countries, and they were introduced at different time points. And the reasons for their import or for their introduction, they were different. Some for the experimental culture, some were for the mosquito controls, and so on. However, out of the different exotic species, the common carp, the silver carp, tilapia, and guppy, these are the common ones which are now of the major concern. So when we talk about Indian rivers, in Ganga, which is the Ganges, and Yamuna rivers, so Nile tilapia and common carp, these are the two fishes, exotic fishes, that they have displaced the native varieties or the native species, which is the Indian major carps. Now, similarly, introduction of the Cyprinus carpio to the Dal Lake, which is one of the most important lake in the heart of Kashmir, and Loktak Lake, which is one of the largest oligotrophic lake in the North India. It has affected the population of the indigenous fish species, that is Schizothorax and Osteobrama. And as a result, their population has declined considerably. Similarly, Gobind Sagar Reservoir, I'll be talking about in detail about the Gobind Sagar Reservoir. The population of Katla and the Mahasir, it got depleted after the introduction of the exotic species, that is the silver carp, in 1971. So this bar graph over here, it gives us an overview of the difference in the various difference in the local and the exotic fishes in the different rivers found in India. Now, so whenever like the quantitation or whenever the estimation of this kind of a data or study related to in developing this data for calculating the number of exotic fishes and the local fishes was desired, then the traditional methods for the data collection included that the people would go to, or students would go to the landing site, or researcher would go to the landing sites, and these landing sites are basically the sites which have been approved by the government of India to actually study the catch. And then for every, the researcher would go there and they would jot down the species name, weigh the fish, and record all the morphometric criteria of the fishes. And then the monthly catch data was calculated. Now, and the conventional methods for catching there were different conventional methods for catching the fishes, like fish pot, angling, push net, and so on. However, there are certain limitations of the conventional methods. The first important limitation is that the species monitoring here, it relies on the physical identification by the visual detection of the morphological characters. So this actually translates into the fact that the person who is actually going to the landing site must be learned enough so that he or she has a proper taxonomic knowledge and should be well versed with the morphological identification or morphological characterization of a particular, of all the fishes or that are present there, or the fish species that they are interested in. Another limitation is that there are certain fish species which are generally, if they are there in the larval stages or during the developing stages, then the probability of detection of such species is comparatively low. So if the researcher or if a person who is going to the landing site is not able to find physically or detect the fish at the landing site physically, then the conclusion would be that the fish is either absent or not present. And this could translate into a kind of a false data, or not it would not represent the actual data. Another limitation is that the whole process is very time consuming, it is very expensive, and it is an invasive method, it may lead to contamination of the ecosystem also. So keeping in view that the limitations of these conventional methods, and keeping also in view that the detection of the species, whether it is native or whether it is exotic, is dependent upon the catch, and which is in turn dependent on the density when it reaches a particular threshold, so that we can get a good amount of catch and therefore interpret the data accordingly. So keeping this limitation in mind, so we know that there is a need to have a non-invasive chief methods where these errors can obviously they cannot be eliminated, but at least they can be minimized. So this graph just gives you an overview of how the eDNA or environmental DNA based sampling is more cost effective whenever the densities, a species density is at low, and how the effort for this kind of a study is comparatively less as compared to that of the physical studies or the conventional studies. So now coming to the case study of freshwater fish diversity in the Gobind Sagar and the Pong Reservoir using eDNA metabarcoding. Okay, so I'll just skip this slide. So the collection sites here, you can see that both the Pong Reservoir as well as the Gobind Sagar reservoirs, these two sampling sites, they are present in the state of Himachal Pradesh. And these are the two regions, or these are the two places which are not very small regions. These are spread over a very long distances. So Gobind Sagar, coming to the Gobind Sagar reservoir. So Gobind Sagar reservoir is located in an area of 15,867 hectares, having a length of 96, almost 97 kilometers, with a width of 6 kilometers in the widest part, and a mean depth of 55 meters. This is a reservoir which is one of the very important source of fish supplies, specifically to the hilly states or the Himalayan states of the region, including Himachal, Punjab. And it is located on the Satluj River. So this is a reservoir which is like a man-made reservoir created on the river Satluj in the foothills of Himachal Pradesh. Similarly, we have a Pong reservoir, which is also known as Maharana Pratap Sagar, named in the honor of late Maharana Pratap, and it is built on the Bias River. So Gobind Sagar is on the Satluj River, and this was on the Bias River. And it is, interestingly, it is one of the international wetland sites which has been declared as under the as one of the Ramsar sites under the Ramsar Convention, and it covers 24,529 hectares with a length of 42 kilometers, width of 19 kilometers in its widest part, and a mean depth of 35.7 meters. So these were the sampling sites, and you can see the distribution of the sampling sites. So the government of India, the government of Himachal Pradesh, carries out routine analysis of the exotic fish species and the local fish species using the conventional approaches. And this graph gives you an overview of the increase, that how over the years till 2015, we can see that there has been an increase in the landing of the exotic fishes in comparison to that of the local fishes. So, and among these different types of exotic fishes, we can see that the percentage of Cyprinus scarpio and the other two fishes is on a higher side or as compared to the others. Similarly, in the Pong reservoir, here we have highlighted the percentage of Cyprinus scarpio. So now coming to our study, so what we tried to study in this in our project is that we wanted to use the eDNA based metabarcoding approach to actually study the fish diversity in both the Pong reservoir as well as the Gobind Sagar reservoir. And also, we know that, as Professor Dirk has already said, and we have talks on the previous day also, that we cannot have the exact abundance of the fishes, but at least we can have some kind of a idea of a relative abundance of the various fishes using the metabarcoding approaches. So in this study, we selected 12S rRNA as one of the target regions to study the fish diversity, and the primers which were used in the study were as used by Valentini et al. In 2016. Although the gene is of 960 base pairs in length, but for metabarcoding purpose, the gene the amplicon size was only 120 to 130 base pairs. So before, as I think in his talk today, Professor Dirk has pointed out that the primer is the most important part in the eDNA metabarcoding. So we also felt the same way, and because that is an important part whenever we are carrying out the metabarcoding studies. So what we did is that before actually going for the high throughput sequencing, we took different types of fishes from the landing sites of these two sampling sites, and we simulated or we extracted the DNA from their tissues, the muscle tissues. And then after the DNA extraction, we created a false sample or a design, or you can say that a sample construct wherein we mixed the DNA of different types of the fishes which are there in the two landing sites. And then after mixing the sample, then we tested the primers that we wanted to use, whether they are able to amplify the different types of fishes from the given pool of DNA. So this we did by carrying out the Sanger sequencing analysis and then followed by the phylogenetic analysis. So once we were satisfied that the primer that we are trying to use is good enough for the study that we want to carry out, we went further and for eDNA metabarcoding. So what we did is that, so we went to the sampling site, and from the sampling site, we collected water sample. So for this purpose, we divided the sampling sites into, so the whole, for example, the Gobind Sagar reservoir. If I talk about Gobind Sagar reservoir, so depending on, you can, I gave you an idea of the width of the Gobind Sagar reservoir. So what we did was that we divide this sampling site into different sub-sampling sites or sampling locations. And then the collection from each of the sampling site, the samples were collected in triplicates. And the samples which were collected in triplicates, they were pooled into one. And then further processed. So what we did is that after collecting the samples, we transported them immediately. And I would tell that they were not brought from Himachal or the Himalayas to the Chandigarh. Chandigarh is a little far away from the sampling site. So what we did is that we had a small substation close by near the sampling site, wherein we took the pooled sample in 4 degrees, and immediately on the same day, we filtered it on a 0.2 micron filter paper. And then this is how it was done. And then after filtration, or after getting the filter paper, then we isolated the DNA from the same. And finally, after extracting the DNA, so DNA was extracted using, we used MP Bio Power Soil Kit for the DNA isolation. And when we isolated the DNA, then the PCR amplification was carried out using the both the specific primers in order to check whether the DNA, that picture, gel picture is not here, by using, in order to check whether the DNA that we had extracted does not have most inhibitors in it, and it should not interfere with the PCR amplification. And then we did it with the universal primer also. And followed by the quantitation of the eDNA using the qubit fluorometer, fluorophore. So finally, so this table here gives you an idea of the different eDNA concentration that we had and that we quantitated using the qubit fluorometer. You can see in this table also that we had 6 sampling sites from the Pong reservoir, and we had 6 sampling sites from the Gobind Sagar reservoir. So why some of the fields, they have been left blank on the last column is because they are still under process, whereas those which are on which the numbers are there, the data for it is in hand, and we are currently partially the data that I'll be showing you today, and we are still in process of deeply analyzing the same. Okay. So what we did is that after, as you know now, that after we had the amplicon, or after during the second round of the PCR, then the adapters were added, the barcodes were added to the samples so as to differentiate between them. And then finally, they were sent for sequencing. And this picture is of a MySeq, but we did not do the sequencing on a MySeq platform. We did it on a HiSeq Extend platform sequencer. And then after carrying out the sequencing on the HiSeq platform, the raw reads were obtained. So again, so this is just an overview of how the whole thing was carried out, and finally the bioinformatic analysis was carried out. So the raw reads which were obtained from the HiSeq sequencing, these raw reads were obviously, they were QC'd. They were the adapters were removed, and the initial they were separated, and then after that they were after the QC analysis. So the database which was used to carry out the analysis of this sample was the MitoFish database. So the MitoFish database is a database which is of the fish mitogenomes, and it is there since 2013, and it is freely available. You can just Google it and see the database. It is generally actively used by in evolutionary biology and ecologists, and all the scientists working in this field use this database very actively. And so this MitoFish and the fish mitogenome, there is a pipeline also called as the MitoAnnotator pipeline, which can be used to annotate the sequencing downstream. So, interestingly, this MitoFish database, it is completely focused on the mitochondrial genome, or that we call as mitogenome of only the fishes, since we know that they are very important in the ecology and in the hydrosphere. So we used this MitoFish database, which is there, and then alignment after with the mitofish database was carried out, and NCBI BLAST was used for the same. And then those reads which could not be aligned to the mitofish database for our analysis, we filtered the unaligned read and then processed for the sequences which were aligned for abundance count and diversity calculations. Now here I would want to point out that there is a possibility that those filtered unaligned read, there's a possibility that the database might not have the sequences, but for the initial study of our data set, we thought to proceed in this manner only. And after we have not yet done this, but we plan to use the filtered unaligned read also to carry out their separate analysis and see what comes out of that. Okay, so this fish, finally the BLAST analysis, you can see that how many reads are there, and out of the BLAST analysis shows that whatever we were interested in, a total of 58 interested fish species were checked, and 40 were observed to be present in both the Pong reservoir as well as the Gobind Sagar reservoir. If we talk about the alpha diversity between the two, we unfortunately we right now have only 2 samples from the Pong reservoir, and we have all the 6 samples from the Gobind Sagar reservoir. So we can see that the alpha diversity was more in the Gobind Sagar reservoir as compared to that of the Pong reservoir. Similarly, the beta diversity analysis, it shows that again, the diversity in the Gobind Sagar reservoir is more as compared to that of the Pong reservoir. Now coming to this pie charts, these pie charts might look a little busy, but I would still want to focus on this, is that, so this pie charts, basically they are showing the relative abundance of the fishes, and you can see that these two are the Pong samples, for which I have only the 2 data sets only for this sampling site. And you can see that here Sperata singhala is one of the most important or one of the dominant. I cannot say that not in terms of the abundance, but we can say that in a rough estimate of kind of a relative abundance. So in a rough estimate, you can see that the Sperata singhala is it represents one of the most dominant fishes as compared to that of the other species. And interestingly, Sperata singhala is a native species. Okay, so as compared to that of the other species, you can see here that we have silver carp and we have Cyprinus carpio, the common carp also. They were also detected, but their number is very low. So in Pong reservoir, we can say that the major fish which is playing the role in maintaining the overall physiology of the reservoir ecosystem is Sperata singhala, and followed by Parambasis and Zenotodon. So these are all native fishes. So this is a very interesting observation, which is different from what we were having from the data that was collected by officially by using the traditional methods. Similarly, now coming to the Gobind Sagar reservoir. So we had 6 different sampling sites for the Gobind Sagar reservoir. So we can see that in the Gobind Sagar reservoir, now this the silver carp was very dominant, and you can see that after the silver carp in sample number 7, S7, you can see that the silver carp was also there. So Cyprinus carpio was also there. So it has been found that the studies have shown that the places where the common carp, which is the Cyprinus carpio, and the silver carp, which is hypothalamic these. So the common carp is dominant generally in the sites where the water quality is not very good. So it is, which means that it is able to survive under the conditions where the water has a very high turbidity and the nutrient content is on the higher side. It is also able to withstand high temperature and also the slew of pollutants and the agricultural runoff. So similarly, the silver carp also, it has been found to be very useful in the algal blooms. Now, there were other fishes. You can see in, there were other fishes like Botia, Sphori. These are the fishes which could not be detected by the conventional methods, but they were detected in the Gobind Sagar Reservoir using this, using the eDNA metabarcoding. Similarly, in the other two sites, that is the site 9 and site 10, again, we can see that the Spiretta singhala and the Cyprinus carpio, these are the two fishes which were dominant, the common carp and the common carp is there. And then similarly in the site Lithani, so there was an eel, which is a very fast moving eel, and they feed on the insect larvae that was detected. Interestingly, this eel was never detected by any of the traditional cultivation methods by the people who were carrying out sampling, who were sampling these sites. So, in the Gobind Sagar Reservoir, there were few endemic species also which were in good competition to the exotic species. Similarly, in site 11 and site 12, we can see, so till here in these sites, that is site 7, site 8, and 9 and 10, we can see that there are only the exotic species which are more dominant as compared to that of the indigenous species. But in site 11 and 12, like that of the Pong Reservoir, what we found that the Spiretta singhala, which is the indigenous variety, is more dominant as compared to the exotic species. Now, the interpretation of this could be many. The first thing, though we had taken good care to take 3, sometimes even we took more than 3 replicates from the sampling site, that was taken, and the DNA extraction was done very carefully also. But still there is a possibility that there can be, several hypotheses can be there. The first that we can generalize is that actually there is in the different sampling sites within the Gobind Sagar Reservoir that there is different distribution of the indigenous and the exotic fishes within the reservoir itself. So that there is a kind of small sub-ecosystem within the reservoir wherein the different fishes have kind of adapted themselves and made their own niche wherein they can live freely without having without competing with each other for the food and space. The second thing could be that if we take it as a technical error also, that there is a possibility that the sample, there is a possibility that we could have actually sampling or technical, the replicates were not enough, and from the same sampling site we could have done more sequencing. So we have actually sequenced, even though we had collected so many samples from the same sampling site, but we have sequenced only one from one sampling site. So another thing could be that we could, from one sampling site, we can increase the number of sequencing replicate, though it is very costly and it will be very expensive for us. But I think that another thing which can be done, if there is some error during the PCR amplification, or there is some that there was some kind of a bias. So in order to overcome that aspect, that can be done. So that is one thing which is still open, and we would want to look into it in future. So we can conclude from this study is that majority of the dominant species found in the samples, both in the specifically in the Govind Sagar reservoir, they were exotic. However, exotic species were also detected in the Pong reservoir. Now, and the number of the endemic or the native species, they were comparatively, I cannot say that the abundance because it is till we carry out the qPCR analysis that we plan to do. So it would not be fair enough to conclude from this preliminary analysis that the abundance of the exotic species is more as compared to that of the endemic species. The only thing that we can see that the number of reads which have gone into during the analysis or the bioinformatic analysis for the exotic species is more as compared to that of the endemic species. Little bit of more statistical analysis is still required or still desired. So, in nutshell, we can say that yes, the introduction of the common carp or the exotic species has affected the indigenous population to a greater extent. Yes, they have, and our results are in agreement with those of the results which are obtained from the landing data. But the advantage is that a number of species which could not be detected by the conventional methods, they are detected by using the DNA metabarcoding approach. So this slide gives you an overview of the other research projects which are going on in our lab. One of which is the estimation of the biodiversity of the hypersaline Trans-Himalayan lake, which I had wished to show the data. I had data, but since it was a microbial data, so I did not want to, I wanted to stick to only the eDNA metabarcoding data related to the vertebrate system only. Another research project, it studies the permafrost affected, not permafrost, permafrost affected soil biodiversity in again the same Changthang region of Ladakh. And we are also studying the, by using eDNA metabarcoding, the biodiversity which is there in the sediment of the hypersaline lake. Similar thing in the second phase, we are planning to carry out for this Gobind Sagar reservoir and the Pong Dam also. The reason for this is because, as also pointed out by Dr. Dirk, is that the stability of the environmental DNA is more in the sediments as compared to that of the water. Though our water system is not that flowing as the rivers, but still there is a little bit of movement is there. But we think that the fish species that we are a little doubtful about, or those which could not be detected, they could be detected in the sediments, and we can have a comparison between what we got in the water and what we would be having in the sediment samples. So that is the future. That is one of the future prospects that we plan to carry out. So all this research is not done without the effort of our so dear students which are there in our lab. So the funding from the DST SERV and the UGC and the hard work that has gone behind this, and all the research scholars which we are working in my lab, Anuragini, Dolma, Ali, and Namita. I thank you for all their efforts for carrying out this study. Thank you so much.
Thank you so much, ma'am, for that impressive data you have shown us and shared with us. The questionnaire session will be happening after the last talk. So I'll be putting forth the questions to you after the talk by Dr. Dirk. So without delaying much, I'll call Dr. Dirk to give his last presentation for the session.
Yep.
Dr. Dirk.
I can still only the
Yeah.
other screen. So are you?
Archana ma'am, can you
Yes.
please stop here?
Yes, yes, yes.
Okay.
Give me a minute.
Yeah.
Okay.
Thank you. Thank you.
Here we go. I'll do the same thing as I did before. Let me just switch here. So you should be able to see my little presentation.
Yes.
Perfect. So this last talk is really trying to give you an overview of what's going on, which is absolutely impossible in whatever time somebody is given. So as much as a PCR is biased, I'm biased towards my own research or things that are probably I was at least remotely involved in, but I'm trying to give an overview of a few things in general everywhere else. So what we're looking at, I'm not sure, yeah, okay, is examples for metabarcoding use, examples from how we use eDNA and everything which is generally probably falling under the category biodiversity genomics, which is really just using genetic tools to get a better idea of biodiversity around us. And that goes from the simple species identification. DNA barcoding aims at probably looking at the interactions between species, functional diversity, and a lot more things. And the more we get into these methods, and the more we also consider probably including full genomic analysis and everything else, you could think about how far this field can go. So you can place an organism not only at a point in time, where it is, where it was. You can make predictions about distributions. You can also start to think about what kind of functional consequences that has for the individual, metabolically and not, and things like that. So biodiversity genomics encompasses pretty much all of that. And I can give you only one angle of it, which is where we are at, and many others. I'm not saying there's not other aspects to it, but sort of in the interest of time. And I know I was already too long in my first presentation this morning. So a couple of examples, and I like to start with that slide, which is the main slogan for the International Barcode of Life project. This consortium of over 30 nations that has this has several goals, but one of them is really just increase the amount of species available for our general reference libraries to up to 2.5 million of them. Also looking more into relations between species, because they've used a term that is not new, but it's called symbiome, which is essentially an organism and all the universe it's carrying with it. So inside as internal symbionts, internal parasites, outside like ectoparasites and everything on them, commensals. So they're also trying to get a better, use the methods we've been already using to get insights into that. So that's one thing that's currently going on. One part of that is one study or one enterprise we're doing in Canada, but there's sort of copies in other participating nations as well. And this is called TRACE, or Tracking the Response of Arthropod Communities to Changing Environments. So what you're seeing here is a larger scale survey type of analysis. So the idea is that we're looking at terrestrial arthropods. We're looking at one particular method to catch them, which is a malaise trap, these tent-like structures you see there, because they're very effective, low maintenance, and so on. We have lots of experience with them. And now we're placing them into different areas. And the one thing that was sort of, and you'll see that in a few more slides, playing a big role is how to select these. How do you possibly cover all the biodiversity in a particular regions? And given that there is a lot of different areas on this planet and a lot of areas where we know they're biodiversity hotspots, so how do you collect there as opposed to probably a temperate region like here in Canada? And like that. So, and one thing we settled on, and I'll talk about that in a few slides now, is ecoregions. Definitions of areas that have common geographical and ecological features. There's a lot of abiotic and biotic factors they share in common, which means they also, and it's expressed in a way that you find similar communities within this particular region. there's no country boundaries that play a role. Ecoregions just are usually shaped by geological features, by habitat differences, and so on. So we use those as a starting point because we cannot impossibly survey the entire planet as dense as we would love to in order to get every information out of it. So trace is one approach to that. So you pick these traps, you replicate them, several of them across different ecoregions of interest. And then here it's sort of as a bifurcating process. We do bulk sampling, we do metabarcoding with these samples, but then also we take an extract of that, especially from things that we obtained in the metabarcoding process that were new to us, that are subjected into our other high throughput based, but still traditional DNA barcoding routine, so that we can at the same time observe an entire community, probably compare it to other samples we had already in place. So this is also going forward in time, but also then whenever we obtain something that we hadn't seen yet before, we can go back and move along the traditional way to extend our reference library. So here in Canada, the metabarcoding part is probably stronger because we've done so much groundwork. But in other areas of the planet, we work closely with Costa Rica, for example, with Dan Jensen. And there in his several tropical forest areas, you put a Malay strap there and pretty much everything is not in our reference library yet, so we're slowly building that up. So we will sort through all these individual ones, and then it goes to the high throughput thing we have there. So this is trace. This is part of an integral part of eyeball and how it does things. There's more than trace. It's just one aspect of it, how we do things and how we're trying to organize large-scale sampling efforts. Here's a bit of how this procedure works. It's just a different view on how a metabarcoding routine functions. So we've seen that in the metabarcoding talk lecture I've given yesterday. So it's really just away from an insect, bottle full of insects with ethanol. We subject it to lysis. We take only the lysate out of it, keep the insects intact as much as we want to, and then go all the way through to sequencing. In this case, if we want to do it fast, we use an S5 approach. So that's one study we've done initially in order to find out if ecoregions are really something that makes sense in the context of our barcoding work. And this is actually looking at 3 different ecoregions in the region I live in. And if you can see, I don't know if you can see my mouse, so somewhere pretty much in the center, close to Toronto, is where Guelph is, so which is where I'm currently at. And there's 3 major ecoregions here. So in terms of variability, it's a pretty boring area. And quite honestly, the one you see here in red is largely now dominated not necessarily by natural features, but rather by agriculture and its consequences, because I think in Southern Ontario, we don't have any primary vegetation left. It's all secondary, regrowth, whatever, if you find it, and the rest is really just agriculture. So it's a largely human-dominated landscape. And we were looking at, can our data, can the composition of species we find with metabarcoding tell a difference between these ecoregions? So essentially, if I get a community from one ecoregion, is it different from the other ecoregion? Or is it more closely related to something that's within the same ecoregion, or within something that is probably in a different one, but very close by? So does geographic distance play a larger role, or does the separation into these regions, which are man-made ideas, but still, does that make more sense? And we saw clearly that there is not a strong signal between geographic distance and diversity. You could find traps that are very close to each other, but there could be very different communities to sample with them, while on the other hand, you could have some that are further apart from each other and they're very similar. So distance is not the shaping mechanism, but ecoregions seem to be on. You see that in the graphs here on the right, that there's a very, very low correlation between similarities and distance, but between, for example, phylogenetic distance, so the diversity within a sample, there's a clear difference between the different ecoregions. And this NDMS plot shows you the same way. So we sort of have clusters that are more particular for the ecoregions, which is a good news for us if we're trying to do sampling design. So now we know if we want diversity covered and not so much local ecology, but more like if we design a survey of everything, get as many species in there as possible for our reference library, then we should start with the basic grid, which is an ecoregion, and then smartly distribute our trapping and our collecting events, not only terrestrial arthropods, but others, in a way that follows that. And you can see here, we work quite a bit in getting up to 1 million barcodes, and our rarefaction curve, it doesn't really fully saturate, although we are close to about 25,000 species of terrestrial arthropods here in southern Ontario. Again, that's a temperate region. I'm not sitting in a tropical rainforest where this curve would probably look dramatically different. And still, although we've put a lot of work in there, our curve is not going, it's slowly saturating, meaning we're close to the completeness, at least for this part of life. So that's one example how we do things and how we use metabarcoding and new ways of DNA barcoding to actually build our reference library and see changes over time. Another thing we use the high throughput sequencing methods for is a totally different one, because over years when we did barcoding, we had thought about, okay, the real treasure trove for building reference material are our museums, our collections. But very often that was hindered by the fact that DNA degrades over time, meaning a specimen, even if it's not kept in awful chemical conditions like formalin or so, over time DNA degrades. So if a specimen is very old, the chance of getting a full-length barcode is relatively low if you do it the regular way. Again, I'm not talking about formalin preserved material, because that's usually, if not properly buffered and everything, it's lost for any kind of DNA analysis, unfortunately. We haven't found any way of obtaining DNA in a reasonable way. Sometimes it works, but it's very rare and then also very random. But I'm talking about large scale, for example, insect collections that have pinned dried specimens, and dry conditions are usually good for DNA. It keeps it preserved for a longer time. So here's an approach using the high throughput sequencing, some next-gen methods, in order to get a full-length reference barcode out of a museum specimen. We've done that successfully for some that are 150 years old. What you do is you design a series of primers that are scattered along the fragment, the CO1 fragment. So it's a combination of a few dozen primers that deliver several aspects or several shorter fragments, which you later on just put together in one contact, which represents your full-length barcode. We could do that initially with Sanger sequencing, but it's just a way too elaborate process to do that. But with next generation or high throughput sequencing, we had the amount of fragments we needed. And now we can do that on a regular basis. So people are sending us museum material regularly, and we just go through them and obtain DNA barcodes from the best possible specimen, a type, which is ideal for whatever reference you're going to build. The only thing, or the other thing, the extra thing you can see, and when we switched from one HTS method, initially S5 and MySeq, to the long read smart sequencing on the PacBio, we started to also observe some patterns that were just simply interesting. As I said, DNA degrades over time, and in these museum specimens it does. And when one of our PhD students looked at Lepidoptera and how they behaved when they were in a collection longer, he was seeing not only the usual trajectory, which you see on the left, that over time DNA gets shorter, but he also observed that there is a couple of nucleotide misincorporations. That means you have a certain propensity for errors occurring in the sequences depending on the age. So the decay leads to something that happens in the sequencing process, that all of a sudden you obtain the wrong nucleotide information. And there's a likelihood that is higher for certain misincorporations than others, which was an interesting find. So there's actually throughout the decay process things going on that lead to certain errors that you later on in sequencing can obtain. Another big topic where there's lots of studies out there, and probably the most when it comes to metabarcoding and eDNA, is certainly biomonitoring. It's the idea that you can use metabarcoding, especially combined with eDNA or with bulk sampling, to do biomonitoring. Some people call it biomonitoring 2.0, whatever you want to coin it, but it is certainly one of the future aspects in how you can do biomonitoring in a totally different way than we are used to do it. Because regularly it's really just looking morphologically at individual specimens. And here are just some examples. There's plenty out there. If you screen the literature for it, you have a lot of reading to do, I promise you. One thing which we had recently, because it popped in my head first when I did this presentation, simply because the paper was just published, and it's kind of, oh yeah, why not talking about it very quickly? So we looked at riparian forests, so forests that are enclosed on rivers that provide shade and everything else there. And especially we were interested in the riparian forests that are close to rivers that are influenced by agricultural runoff. So that upstream somewhere there is agricultural business going on. And of course, there's a huge influence an influx of everything, of nutrients, of pesticides, of sediment, you name it. And we were looking at two different river systems, so examples for both of them. One of them had no forests around them anymore because humans had removed them, because it's a heavily populated area or whatever. And others where the riparian forest was there. And we found clear differences, especially for some of the ecological factors we looked at. So we did metabarcoding of the communities, looked at the different compositions of it, and correlated them with a lot of environmental factors. And what came out very strongly is the role that riparian forests can play, especially now in times of global warming. We know we have an increase in temperature. We will observe that for a longer while. And some of the species within the freshwater body, and many of you will know that, are very, very sensitive to changes in temperature and others. And we could clearly show that the presence of a riparian forest, a forest around the river, provides shade for some of the temperature-sensitive species and everything else. So the survival of them is probably much higher if you have any kind of these natural environments still present there. So a forest like that can mitigate warming and ecological degradation, because that's what we also were able to show. If you have them present, the amount of ecological degradation, even with an upstream activity of humans in agriculture, is it's much better buffered than it would be without. Other examples is that you can actually go further with metabarcoding than I said before. So far I was only talking about assigning species names, coming up with species community lists and all that, but you can go further if you play, and you can see that here in the middle, if you play with the bioinformatics, if you are starting to filter stricter than you normally would do, if you really make sure that the likelihood of an error popping in is low, you will reduce your species list to a couple of them, not a couple, but way less than you normally would have. However, you would also have way more individual data to particular species. So you would pick up species for which you have several individuals in your samples. And then if you have samples across a certain area, so from all the individual samples, you can have contributing factors. Now you can start to see population differences. Now you can look at the interspecific genetic diversity in a particular area. So we're looking all of a sudden at the opportunity that a metabarcoding dataset, initially meant to provide you with a community list, provides you even more information. You can start building haplotype networks, as you can see them in the slide, for individual species. Yes, you've started with 20 million sequences out of a MySeq run, and you have reduced it probably just to a few dozen species where you have enough information, but still, you can do it. And now imagine when your input is a more modern method that produces billions of sequences. Again, you go back and the amount of information you get out of it is much larger. So these kind of data we're already producing have the power to give you way more information than just the normal purpose we're using them for. I'm not saying this is a bad one. The OTU or species lists are highly valuable, But imagine what you can do when you have some sort of population differences. Mind you, that's only possible when your marker of choice for metabarcoding actually allows to do that and provide that. That depends a little on the species and the marker choice. But if you are after that, that's one way of doing it. And we showed, and a couple of others did that too, that it's possible. We also looked at community restoration along farm fields. This is something, a very similar process. You've seen that before. We use the same protocol. What we're doing is having traps on farms, regular farms, but with one little difference. So we have regular, real large-scale operations, but also smaller farms, a few of the ones that do more, environmentally oriented farming. But then one thing is important for many of the mid-sized farms that are very typical for Southern Ontario. These are people that came together in an organization that has one goal, that they use marginal land, so areas of the field that they are normally not used for farming because it's just not feasible. They leave them and they allow that the original flora and fauna comes back. So it's a restoration essentially that happens there. And we were looking at whether there's an influence of having a small patch of land on a farm with these natural surroundings. So here in the area is what we call tallgrass prairie, which is very typical for where these farms are. And we looked at the effect of them. And it turns out it has a huge one. Arthropod communities thrive even on an agricultural enterprise when you have small patches of natural occurring land, land left alone. Yes, there will be influx from artificial nutrition, from pesticide use and everything else. Still, small patches are already a big step forward towards a better. We all know that agriculture is probably the most problematic thing when you think about biodiversity and how it's threatened. The loss of species, the fragmentation of the landscape, and everything has to do with it, even climate change to some extent. Agriculture is always identified as the main culprit, among other things, but usually at the top of the list. If you find methods of reducing the impact of agriculture, because we need it, we need to eat, so we are looking into ways of probably just live with both at a proper way. There's one indication that this kind of farming has a better future than the large-scale industrialized barren land approach. That's unfortunately in North America very common too. So we were able to show that these patches of land actually have a positive effect on the arthropod community. One thing I usually like to point out in these kinds of talks is what you can do with the data and how far you can actually drive that. Yes, we come up with a species list, so eventually we can probably show how a community in a particular point is. But now imagine how much other data is out there. We get more and more of these Earth observation data, so satellite imagery, LiDAR. When we talk about agriculture, there's a lot of these modern tractor technologies where the tractor itself records plenty of information on a square meter scale. So some people who can afford these tractors, that's the one important thing, and not everyone can. They record for every square meter the amount of pesticide they brought out, the amount of fertilizer they brought out, what they planted, in what distance and whatever. These are coming in square meter resolution. And then you take that together with observational data from looking above, like satellite imagery, which comes at high resolution, LiDAR, all that. What's missing is mostly a layer of the diversity that's living with it. And that's a difficult one because you cannot normally observe that very well from above, nor with these tractor data or anything else. So what it needs is this layer of information. Large-scale sampling would provide that, but we are nowhere close to a square meter range. We place a trap, 1 or 2 traps in a certain area, which is covered by all these other data, and then we hope that this is enough. And then we can come up with probably something like what everybody knows for individual species distribution models. So calculations of where species can occur, where it's more likely and less likely based on the information we have at hand. Now you can do joint species distribution modeling, which is sort of taking it up a notch. So it's like coming from the individual barcode to metabarcoding. We go from individual species distribution modeling to the joint one, which means that we are trying to look at community and how they could be distributed and how they vary over a certain area. The problem is we have point samples. We have a collection there, we have a collection there and there. So how do you make a continuous map of that, which is needed if you want to compare it to all the other sources of information? So it's sort of you overlay each of them, one of them, and that's a bit of a problem. We tried it a bit. So we tried modeling. You can see here the diversity, the alpha diversity. And we tried different modeling approaches, and these were agricultural fields where we had 5 Malaise traps and not much, and they were quite distant from each other. And we were trying to see how much information in terms of distribution of community diversity we can find. And you can see some of the heat maps there really provide you a nice signal. There's a bit of uncertainty in there, but this is the reality. This is how many traps you can probably put in there. And if you think downstream of the sequencing analysis, it's also what your budget might allow you to do. So you have to come up with smart ways of modeling that, and it seems to work. And now you have this information. So you can overlay diversity, overlay with tractor data. Some of our agricultural specialists and the economists have sat down there and said, okay, what does it cost a farmer for his entire operation, but then also break it down to the individual square meter and said, okay, what part of your field is actually making money and where do you lose money? And with the tractor data and all the other informations they had at hand, it's usually yield and what you put in there and what you pay for the field and everything else. They had a clear understanding of, okay, what's the profitable part of a farm and what's the less profitable one. Now we throw in our biodiversity layer, and we can probably, we're starting to look at correlations there, tell them if biodiversity, if it's richer at a certain area because of certain features around it, does it make the field more profitable than one where there's low biodiversity? And this is what we're after with this kind of mapping, when you think about it from a more practical approach to it. But there's lots of different applications. Very quickly, another thing we're doing here for years, we try to work with the youngest citizen science, with school kids, in order to help inform some of the ecological studies we're doing. So one famous project right now is the bees at school. So the idea is, you see here, artificial nesting structures for bees that put their eggs into holes in trees and whatever. And you can provide these nesting structures. There's famous bee hotels. You can actually buy them commercially pretty much everywhere and put them out to help these kinds of bees. They're usually solitary bees, not talking about honeybees or anything of that sort. The idea is that we try to understand these animals better, try to know who's where. And in order to do that, you have to sort of spread out your collecting. And what's better than saying, hey, schools, would you be willing to put one of those out throughout the entire summer season? The kids have a chance to observe what's going on. We will take them back. We use metabarcoding to identify what the larvae are that are in these tubes. And you see an open tube there. Number two, very many of these bees will actually put a layer of leaves or any kind of plant material around inside the tubes, like tapestry or so, they put in there as protection. So we can use metabarcoding, we do, to determine what these species are. So we know not only what bees are around there, we know what they're using for this. And we take the pollen that they usually provision for their larvae. So the mother usually leaves a portion of pollen behind for the larvae. And the larvae have started eating it already, so we obtain it through the gut content of the larvae and the remaining pollen. So we know what food source they were using. So now we have all these information. We know who is there, what have they been eating, and what have they been utilizing for building the nesting structure, which gives us a very clear picture for this individual species. And now we're distributing it across Canada, 200 schools at a time. Very successful, even in COVID lockdown times. We still had a lot of schools and teachers participating. They actually loved the idea of doing something else than sitting at home and doing online learning. So that's why this was probably so successful last year, and we just got funding to continue for 3 more years. So now over time, we will build this wonderful data set of all these different layers of information, and we really get a complete picture of what's going on there. And we've been looking at pollen all the time. Pollen, eDNA, Metabarcoding, you name it. This is just a snapshot of a study where we looked at non-bee pollinators, where a student actually went through the exercise of washing pollen off every individual insect she's collected, and then looked at the distribution of plants, and clearly saw differences between some species that utilize pretty much everything that flowers around them, and others that are highly specialized on them. And she could also show, for example, that yes, a honeybee is a very specialized pollen collector. It can carry it home and everything else. So per bee, you have a lot of pollen, which makes people think that they are probably the most important part of the entire pollination thing. However, you're looking at flies, syrphid flies in particular. They might not carry a lot of pollen on them because they have no particular apparatus or anything on their body, but they carry it all around there. And they are equally, because they're doing it with a higher frequency, with a higher number of individuals, they're probably contributing the same amount as non-bees to the pollination services as the bees themselves would do. Bigger thing, lots of people started doing ever since we had high throughput sequencing out there, and even before. Barcoding was a good way of doing so. Dietary analysis using DNA is an old thing. But with high throughput technology, it became really, really interesting, and we were really able to do a lot of interesting things with it. And I just browsed through a few. One is a more human thing. It's a few years ago that I was approached by people that are interested in the microbiome of people. So in our gut microbiome, they wanted to know, is there differences between different human populations and different human groups for their microbiome? And of course there is. And they had picked on a particular group of people because it's thought that these people, the Hadza in Tanzania, have one of the most ancient lifestyles we can imagine, because they are not farmers. They don't have livestock. They live completely opportunistic. So they're hunters and gatherers still. There's only 200 of them remaining in a small area there. And they're trying to keep their ways of living up. And this is a perfect sort of null hypothesis, and said, okay, we think that humans, in particular in this region, used to live like that, and not like us, having a different type of food sources and everything else. And we clearly saw that. The microbiomes is so different. You see that here in this figure at the lower part, where you have these histograms on the side. And at the lowest level is all these civilized countries, like whatever, North America, Europe, some parts of Asia, where the diet has shaped the microbiome community quite differently from all the others, which are usually smaller tribes, people, Native Americans in South America, and then people in Africa and in some remote areas of Asia, where you see clear differences. It's a total different community in their guts. Although we're all human, we once shared all those. So it's not so much, and we know that the food available to people shapes that. So when looking at the Hadza and using fecal samples to look at the microbiomes, first they discovered that actually there's seasonality in there, strongly dependent on rain season and dry season and the availability of food there. And the microbiome shifts quite dramatically within these two, with these two seasons. The one thing we didn't know initially was, what have the Hadza been actually eating? Because you can only do that by surveying them. And if you're a hunter and gatherer with a huge food supply, you usually don't keep track of what you've been eating a couple of days ago. And it takes a while until the food passes through your gut. And even if I would ask you, what did you had for dinner 3 days ago? You might recall what it was. And then I would ask you what species were in there. Then it becomes rather difficult. So we used metabarcoding. We used the fecal samples, the same origin as the microbiome studies, and looked at the fecal samples. And here's the spread of what they actually were eating. We know they have over 600 species in their menu. Just to give you an idea, the average North American, which is probably the poorest diet you can think of, if you're lucky, you get 10 to 15. So this is a huge difference, and it has an influence. And also the fact that they have a strong plant-based diet with a lot of tubers, with a lot of material that requires cellulose digestion, which normally humans can't do because we don't have the repertoire of bacteria in our guts. But there are some groups there that can do it. So there is a bit of more usage of food that I, for example, if I were to eat it, it would pass through as fiber. That's it. But they have a strong fiber-based diet, so their microbiome is actually capable of using some of it. Not as effective as an ungulator, but still. So quickly, other dietary studies, because I'm getting towards using too much of my time. So we looked at this wonderful bird, Arachlorhynchus, the red-green macaw, rewilded in Argentina. So it was because of farming and everything else, disappeared over the entire north of Argentina. And then some rewilding, so some reforestation and everything occurred, and then people started thinking about rewilding. So they're now introducing birds bred in captivity out in the wild. But bird in captivity, probably too long or for all its life. So all these birds are not coming from natural population. They're coming from zoos, private people and everything else. So they have to be trained in order to survive, find the right food. And the question was, can these birds actually live for themselves? Will they find back to the old ways of a very generalist diet that allows them to survive throughout the year? And what we did was looking at their diet using a metabarcoding approach to see how much does the diet start going into breadth from the point where they were in large cages and trained and still supply fed, and then to the point where they start removing more and more of the food they provided to them in order to encourage them to go out and forage. And it seems these birds doing quite fine. They're very creative about their food sources, and it seems to provide the right amount of nutrition to them. So there's hope for this population, for these birds to actually be reintroduced and completely rebuild the population in that particular part. We did similar gut gut content studies for crabs, green crab, invasive species, pretty much all around the world right now. Nobody wants them, but they're here at the southern tip of the Americas. They have a problem with them, but they had no idea what they'd be feeding on. So this study sort of enlightens that a bit and gives an idea of what these crabs have been feeding on, and it's a lot. They still have this sort of specialties, so they prefer mussels that they can crack open and all that, but you can also show that because they occur closer to the coastline, a lot of the coast is relatively flat and there's inland winds transporting a lot of material and debris into the ocean. They also eat, you find a lot of terrestrial insects in their guts simply because they were just drifted in the sea and they feed on everything, which is a concerning message. An invasive species that is generalist in its feeding behavior is always a concern. eDNA. There's a few things I would like to say about eDNA. Just 2 more slides, and then I'm pretty much done with that one. eDNA as such, not too much, there's a few slides. We talked about that in the first lecture today. What we were doing is, and that's one of the things where I said, yeah, this is a different way of looking at the entire thing, is we obtain water samples through citizen scientists, but we're not looking for a waterborne organism like a fish or any kind of macro invertebrate. No, we're looking for birds, for water birds that land in these lakes here across Canada, and here you see the pathways of the migration. So we really want to see how they're doing. And then in the next step, we'd like to correlate that with avian influenza dynamics, because the idea is that waterfowl is usually the transporting mechanism for avian influenza. And as soon as these waterfowl, like ducks or geese, come close to a farm that has chicken and everything else, and the avian influenza can jump across the species barrier. So what we're trying to see is, does migration does the bird migration reflect the pathway of avian influenza? So we're looking for both with the eDNA methods. And then here we have developed some methods of using our Malay stripe samples more than just getting the insects out. We take the ethanol, filter it properly, and extract the pollen that's in there. So the pollen that the insects actively carried in there. We cannot assign it to particular species, but we have a very clear understanding of how far it ranges of what we catch and how far these insects go and collect plants. And then, as I mentioned that in my metabarcoding talk, we're also looking at the multi-kingdom communities and trying to use eDNA and eRNA in order to probably inform about entire community compositions. That goes beyond metabarcoding into the metagenomics and metatranscriptomics pathway, and trying to find way. And if that works, then go on further and said, can we actually look at stressor response predictions using these kinds of data set? There's a lot of machine learning that plays into that as well. So this is more like the future down the road. And I know there's a lot of people working on these kind of approaches as well. So as I said, that's the next step. When metabarcoding becomes boring, then people switch probably over to metagenomics and metatranscriptomics. It's also depending on how the technology evolves further and makes things accessible, aka cheaper. And that is my talk. I thank everyone who funds my research, everyone I'm working with, my little group, our institute, the Center for Biodiversity Genomics, of course, the University of Guelph, and everybody who provides funding, including the Shastri Institute, for this lecture series. Thank you.
Thank you, sirs, for your remarkable presentation. I think we can move forward to the questionnaire session to the speakers. So I will just read out the question to the speakers with their name. You can answer one by one. So the first question would be to Dr. Archana. The question is, how does challenges like strong current and huge habitats resulting in dispersal of DNA fragments affect the sampling of eDNA? This is from Dr. Reena Das.
Yes, ma'am. This is a very right question. That yes, as I think it was pointed out by Dr. Stenke also, that yes, the water currents, they do affect the dispersal of the eDNA. And it is possible that the eDNA could have been generated at a particular place, and then where it is, it could have been translocated to an entirely different location. So that is very, very possible. So coming to what I had presented in my reservoir in Pong and in Gobind Sagar, even though they are built on the Satluj and Beas River, but they are not built on the very rapid current terrain, and like it is a kind of a flat region. And what can be generalized is that it is possible that from the uphill regions, there could be certain eDNA which is long lasting enough, might reach into the reservoir and can be part of the eDNA that we are extracting. So that is, yes, there are challenges with respect to that, but specifically if we are working in river ecosystem, as also pointed out by Professor Stenke, that yes, in those moving water conditions, it will be difficult to actually find out exactly trap the eDNA which is there, because it is moving. So it is better to sample from a site which is along the river line, but it is more of the movement is less frequent and it is more static. So there the probability of finding the eDNA from the surrounding species, it will be, or which is inhabiting that particular aquatic environment, is more as compared to if you are trying to do it from a runoff water.
Okay, thank you. I think that answers your question. So the next question would be to Dr. Archana again. Can you please elaborate on abundance count and diversity calculation?
This is a very elaborate question, and there can be a full-fledged lecture on this only. But to sum up or to summarize, or in one or two liner, I would just say that, so we have different indices by which we can calculate the species richness, and we can calculate the species evenness. So like we have Simpson, we have Shannon indices. So these are the indices which are used to calculate the species richness and evenness respectively. And what happens is that a generalized methodology which is followed is that typically there is a scale ranging from 0 to 1. When we say it is 0, 0 indicates it is there is very low evenness. And when we say the scale of 1, it indicates there is a high species, single species dominance. So somewhere there has to be the numbers, they range between 0 to 1. So it is, and then we have, if you want to calculate the abundance, we have Chow indices. So I think there is a, you can contact me, I think after this talk also, when we can discuss about these abundant calculations in details.
Okay, ma'am. Thank you. Thank you so much. So the next question is from, no, it's a comment from Mudassir Hakim from Faculty of Veterinary Sciences. Thank you, ma'am. Your work is interesting. You mentioned the fish, Botia, is Botia birdi. The fish is indigenous to Kashmir waters. It is surprising to know the fish has managed to enter warmer waters. What is your comment, ma'am?
So my comment on that, so this is a very good observation, Mudassir. Dr. Mudassir, but I would want to tell you that the fish Botia birdi is, even though it is indigenous to the Kashmir waters, but has been reported, well documented, that it has been found in the Gobind Sagar and the Pong reservoir. It is there. Not only it is Botia birdi, but Botia dario. Then we have Botia lachata, and there is one more species of Botia which is found in these waters. So yes, they are present.
Okay, thank you. So the next question is from Aradhana Menon to Dr. Dirk. What is the minimum eDNA concentration required for barcoding studies?
Well, depends. You're in a good range when you have at least double-digit nanogram per microliter. So I think I remember a table seeing that in today, so where they were in perfect condition. I'd say when we work, we get concerned when it's falling below 5 nanogram per microliter. It's still possible, but then we would probably go to some measures like putting everything again on a column, then elute it with a smaller amount, so smaller volume of elution liquid. Or later on, one trick you can do if you have only 2-3 nanogram per microliter of DNA, one trick, well, not trick, but what desperate people do, they do run several PCRs. So you run one PCR, take the product, make it the template for the next one, so that you increase the amount of amplicon there in cases where you don't have enough.
Okay, sir. Thank you so much. So the next question is again from Aradhana Menon to Dr. Archana. Any particular reason why muzzle from dorsal fin was taken for the DNA extraction in the study?
Yeah, this is again a good observation. So what happens is that if you want to take the muscle tissue from any other part of the fish body, you have to sacrifice the fish. So if you want to include non-lethal methods wherein the fish is alive, so dorsal fin, even without sacrificing the fish, you can just take the tissue and do your analysis. So non-lethal, just for that reason.
Okay, thank you, ma'am. The next question is from Dr. Anand Babu to Dr. Archana. It was a nice presentation, ma'am. Is there any other gene available other than 12S rRNA for metabarcoding studies in aquatic species?
Yes, I think this was well covered by Dr. Stenke in his talk. So yes, there are 12S rRNAs, there's a COI gene, cytochrome B, 16S rRNA, NADS, anything can be used. So it depends upon what your question is. So that is the most important. And out of the various genes, 12S rRNA, since it is a mitochondrial gene, it has more abundance. So therefore, this is preferred from 16S rRNA, but it depends entirely what question you are asking. D loop is used, so it depends.
Okay, thank you, ma'am. Thank you. Then we have another question from Arafat to Dr. Archana. What is the detection limit? That is, how many fish need to be present in a stream to shed enough DNA for a positive result?
I actually don't have any concrete answer to this. It does not depend upon how many fish that needs to be present. What the question should be that whatever DNA has been shed into the water body, is it stable enough and we are able to get it and we are able to extract it so that if we can use it for amplification purpose further. So that is important, I think.
Oh, yeah.
Yeah, so Dr. Stenke, please, if you have any other comment, you can comment.
It's the perfect answer to that because it really depends on so many factors. I've just seen recently a study where people looked at eDNA quantities correlated with activity of the fish. So they had fishes in aquaria and looked how active they were or stressed out and how much that had an influence on shedded DNA. So it even depends on how much your fish swim around and how active they are. If the metabolism is higher, you get more eDNA out of it. So there's no answer to that. It could be just one guppy moving fast and you get a signal, and then for some reason you have a huge carp and nothing happens, or you're not getting enough and you're not over the detection limit. It's really a little more tricky than that.
Yep.
Sorry. The next question is how to remove the qPCR inhibitors from eDNA samples. qPCR inhibitors, right?
So
it is to Dr. Dirk.
How to remove the inhibitors? It's like in every PCR. There's a couple of tricks you can do. There's extra chemistry you can throw in. We stabilize our reaction with trehalose. Others use BSA, and I've seen many different ways of doing it. The best thing is actually trying to get rid of them in your extraction and then trying to find a method that does that. You will never get rid of them completely, but at least look at them. It also depends on if you take water samples. We have one river here. We're really interested in the fish diversity, and what we're currently tackling is the fact that it is a low pH environment with a lot of wood debris and everything else, which doesn't necessarily mean that your PCR is working as nice as it should be. So we're looking at different ways of buffering and see if that works fine. It's the same tricks you would be using in a regular PCR to get rid of the inhibitors.
Okay, sir.
And the extraction method determines. It's a very simple one. You probably heard of the problem that plant species share with some animal species is exerting polysaccharides, which are inhibitors for PCR. So you just switch to, so for example, mollusks doesn't do that. So what we essentially do when we have mollusks in the play, we use a plant extraction method because it does the same trick. It binds the polysaccharides.
Thank you so much.
So if you allow me to add things like DMSO also, it also helps in speeding up the PCR reaction. And when it's pH is another factor. So we face the same problem in our hypersaline lakes. The salinity was high and the pH was high. So you have to actually work with the buffering system in order to get the maximum out of what you have. And lastly, yes, correctly said, it depends. For example, we were using the soil DNA isolation kit because it depends. So you have to actually tweak with your sampling system, depending on extraction system, and which gives you best results. So it depends.
Okay. Thank you so much. So I think we are done with the questions here. Dr. Rama, do you have any questions to ask to the speakers?
I don't have questions, but I'm very happy to have Dr. John Reed with us, who is the former president of the Shastri Indo-Canadian Collaboration, Indo-Canadian Institutes with us. So I'll just give a brief of the event to Dr. Reed, introduce him, and then we will have a few comments from Dr. Reed. So Dr. Reed, it's a pleasure to have you with us. The last 2 days, we've been having these lecture series. There have been 6 lectures on aspects of methods which are used to study the biodiversity of animals, monitor them, and look at what the effects are. So Canada is a global leader in studying biodiversity. The lectures, there's been an excellent lecture by Dr. Sudha Rao from India, who spoke about the bioinformatics and databases to enable metabarcoding. And Dr. Archana Chauhan today showed us some aspects of the work that she has been doing to understand the biodiversity of fishes in some areas of India. Dr. Stenke's lectures have been outstanding. He has given 3 lectures in the last 2 days. He gave us a lecture on the foundations of metabarcoding, and he has spoken to us about the difficulties in using eDNA. And the third lecture was an eye-opener. He gave us a peek into the many applications of metabarcoding. And I, for myself, can see at least a few ways, looking forward, how some of the experiments, applications that Dr. Steenke showed can be modified to be used in India. The program that he showed about pollinator ecology, where we can introduce biology to school students, looks like a very interesting project, because in India also we have a network which is called the Atal Innovative Schools, where we have access to students from 6th standard to 12th grade. And they already have a kit which is given to them, but that is mostly on robotics and sensors. It would be wonderful to have aspects where they can study biology and ecology. So yeah, and we had a total of 150 people who had registered for the events, but we had between 30 and 50 people who were there, who actually came and participated. But that 30% rate should not disappoint us, because the quality of interactions based on the questions that we have seen at the end of sessions has been extremely good. And I'm sure this is going to mature into research ideas from the participants. And I do hope that there is a possibility of collaborating with Canadian institutes. So yeah, so I would like to just introduce Professor John Reed, who is a Professor Emeritus of History, St. Mary's University, Nova Scotia, Canada. He's a past president of the Shastri Indo-Canadian Institutes, and has helped and has a lot of experience in seeing the Indo-Canadian research collaborations grow. Professor Reed holds a degree from Oxford University, a BA and an MA from the Memorial University, and a PhD from the University of New Brunswick. He is the former coordinator of the Atlantic Canada Studies at St. Mary's, and is currently the Senior Research Fellow at the Gosbrook Research Institute. He is a Fellow of the Royal Society of Canada, elected in 2004. Dr. Reed's principal teaching and research interests include the history of early modern Northeastern North America, the history of Atlantic Canada, the history of higher education, and the history of sport. Dr. Reed has served on the Council of the Canadian Historical Association and on the editorial board of the Canadian Historical Review, a current board member of 3 historical journals and co-editor of the Acadiensis, Journal of the History of the Atlantic Region, and University of Toronto Press monograph series. Among other international activities, he was the St. Mary's representative to the Canadian Member Council of SICI, and he became the vice president in 2018, and he was the vice president, I think, in 2019. Yeah, thank you very much, and I'm happy to have Dr. Reed share his thoughts on the way forward.
Thank you very much. I'm delighted to have this opportunity, and let me start by saying that it's always a great pleasure to visit the beautiful and fascinating city of Chennai virtually on this occasion, but I would like it to be known that I've been preparing diligently for this by making previous virtual visits to the city. It's well known to my Shastri Institute colleagues that I'm a great lover of the sport of cricket. And so given the current test match series, 2 of the matches having been in Chennai, I've been quite diligent in watching. And let me tell you that from my vantage point in cold and snowy Halifax, Nova Scotia in the month of February, these aerial views of Marina Beach look very appealing indeed. So anyway, it is a pleasure, even virtually, to visit. More substantially, I've been thoroughly enjoying the lectures, the sessions. I have enjoyed those this morning and had a chance to review briefly the ones yesterday. And while, as you've heard, I am a historian by trade, and therefore I don't claim any expertise in the subjects we've been hearing about. Nevertheless, I think for all of us who live on this planet, in the current environmental circumstances we find ourselves, the matter of biodiversity is critically important. And so that is one reason for valuing these sessions so much. And beyond that, some of the more specific insights, whether it is about the vast number of undocumented species that exist, or whether it be the impact of invasive species, or the many other fascinating dimensions that we've been hearing about yesterday and today. Again, it underlines, I think, how the defining and the measurement of these important elements of DNA is such a crucial subject of research. And honestly, it really has been a privilege to be able to listen to the insights of people who are working at the leading edge of such a very, very important field of research and study. Let me also say that I think this event is just, in many ways, the perfect kind of event to be associated with the Shastri Institute. As Dr. Prachi Kaul was pointing out yesterday, the Shastri Institute exists to assist in creating and nurturing academic and particularly research contacts in all disciplines between Canada and India. And very clearly, these wonderful lectures that we've been hearing at this event are an excellent example of making connections between very significant research in both countries, so that this is, I think, as I say, perfect for the Shastri Institute. And as people may know, the Shastri Institute has just very recently celebrated its golden Jubilee. And this event is one of the events that particularly marks that golden Jubilee. And let me say, I think it's just an ideal kind of model for the kind of event that Shastri exists to help in making happen. And also, it's very interesting, I think, to just observe and comment briefly on the communications methods that have gone into this event. We're all, of course, all too aware that the current global pandemic is an awful and tragic sequence of events. And yet, as the saying goes, necessity is the mother of invention. And so through the use of more remote techniques of communication, I think one of the things we've learned is that we are in a position through these means of communication to make these very direct forms of interchange between researchers that we've seen so much of in the past couple of days. So that yet again, I think that this event has been an excellent example of how research can be communicated in these innovative ways to a wide audience, an audience that I know is from, I think as I remember from yesterday, 40 different institutions. And that in itself, given the quality of the presentations, I think is an enormous and very praiseworthy kind of achievement. So I know from my Shastri perspective that I would speak for all my Shastri colleagues in saying just how delighted we are to have taken some role in making this happen. So let me just conclude by congratulating the organizers. A great deal of work goes into the organization, just the making it happen of an event such as this one. To the participants, the lecturers, for their wonderful presentations, and of course for the congratulations too to the very perceptive members of the audience who have enriched the event with their very interesting comments and questions, and of course that have brought equally interesting responses from the participants. So congratulations to all. Thank you very sincerely for my opportunity to be part of this event. And as others have been saying, I'm sure we can look forward very confidently to future collaborations and future insights that will come out of what has been so productively unfolding in these last couple of days. Thank you very much.
Thank you, sir. Thank you, ma'am. I will just announce it's an announcement for the participants. The participants can fill the feedback form that is in the chat box of the Zoom link and the YouTube link. You can just post the feedback form and you will get your certificates directly through your mail. So at this outset, I would like to thank Shastri Indo-Canadian institutions, especially Dr. Prachi Kaul, for giving us this opportunity, without which we would have not been able to do this program successfully. And I would also like to thank the speakers, Dr. Doug, Dr. Sudharao, Dr. Archana, and also Dr. Prachi and Dr. John for being so accommodative with the time schedule of the lecture series. And I thank the participants for their patience and their enthusiasm in attending this lecture series and making speakers encounter with the questions. And it is the moment for me to express my gratitude to my fellow colleagues for their constant support for this program to be successful. I hope that comes to the conclusion of the lecture series. And I hope to meet you all again and in a different occasion and a different topic. So until then, all of you have a great day ahead and a great year ahead. Thank you.
Thank you.